Antibody and evolution. Antibody in our body is a part of the immune system. When we have invading outside material, so-called antigen, we generate the antibody which can bind to the antigen and neutralize those foreign materials. Antibody is generated by B cells. When B cell is differentiated and matured, it forms a plasma cell, and plasma cell make antibody, and this antibody eventually bind to the antigen and neutralize those antigens function. Antibody is a Y-shaped protein. Antibody is immunoglobulin protein, and the molecular weight is approximately 150 kilodalton. And the heavy chain here, the red color and also this blue color is a heavy chain. Heavy chain, each of them is about 50 kilodalton. And the light chain is green color and yellow color. Each of them is about 25 kilodalton. If you look at the antibody structure, upper part is FAB, fragment antigen binding, and lower part is FC, the fragment of a constant region or crystallizable region. Also, antibody has a two heavy chain and two light chain, and antigen binding site is on top, and hinge region is connecting the two strands of a heavy chain. If you look at antibody and antigen together, antibody's binding site on antigen is called as an epitope. In comparison, the binding site on antibody, that is paratope. So paratope bind to epitope. Let's think about the diversity of antibodies. Antibody has variable regions and constant regions. And these variable regions make different kind of antibodies. We know at least our body can generate millions of different kind of antibodies. How can we generate so many different diversities? If you think the proteins are the product of gene in DNA, our total genome size is composed of a little more than 20,000 of genes. That means in principle we can make about 20,000 proteins. Now let's think that antibody is also a protein. Then with only 20,000 genes, how can you make millions of different antibodies? What's the magic? So the question is, how many genes do we have for the diversity of antibody? Let's think about the heavy chain. Heavy chains, the variable site, is composed of three different domains. Those domains, V, D, J, variable diversity joint areas, they are combination of different V's, different D's, and different J's, and they are selected together to make one antibody. Light chain also has a similar structure. Light chains at the variable area is composed of two variable and joint. For heavy chain, we have 51V, 27D, and 6J. So this combination, they can choose randomly any of those V, D, J one by one, and those combination can make 8,262 different kind of heavy chains. Light chain case, 40V and 5J, it makes 200 types of light chain. Now, these two can make fully random combination together. Altogether, this can make almost 1.6 million of different antibodies. Now, let's count how many genes have been used to make this diversity. In heavy chain, there are 51V, 27D, 6J. Altogether, 84 genes. In the light chain, 40V and 5J, 45 genes. If we combine this together, total number of genes is 129. Utilizing this 129, they can make more than 1.6 million different types of antibodies. So this is utilizing the combinatorial power by using limited number of building blocks. By combining those other sets, they can generate a lot of diversities. This is the magic of antibodies diversity. This set of information is inherited from father and mother. So this set is only one set, then maybe we can generate two sets, maybe about 3 million type of antibody sets from the information inherited from parents. Now this is not the end. These germline antibodies, when they get matured through hypermutations, they change those points by point mutation and then make more matured somatic antibody. So there are a lot more diversity can be generated through this process. Germline antibody is a starting point. Usually they have a flexible conformation and it can bind to many different kinds of antigens. It's very similar to our hand, can bind to many different things. Maybe the binding affinity is not very strong, but it has a flexibility to many different kind of antigen. So this is very similar to induced fit model of an enzyme to substrate binding. In contrast, the somatic antibody, they fix those conformation by point mutation, and then they have a very high specificity and high affinity to the specific antigen, and this is more similar to lock and key model. So germline antibody has millions of diversity inherited from the parent's genetic information, and somatic hypermutations increase those diversity further and further. So altogether, we may have tens of hundreds of millions the different antibody set we can generate. Binding mode of antibody, one is a key and lock, and the other is induced fit. Now you can designate which one is which, right? How to make a monoclonal antibody? If you inject antigen into animal body, then the mouse can generate millions of different antibody, and we can isolate those B cell. Now here the plasma cells, mature B cell, and then hybridize it together with the myeloma cells. These are kind of a cancer cell, and they can make these spleen cells the immortal. So this hybridoma we can niche and then find out the best binding antibody and then select those cell line, and then we can generate single antibody with the highest affinity. This is the way how to make the monoclonal antibody. Single chain FV. Antibody has two arms, and both arms has the same structure, but those binding motif is one part is in heavy chain, the other part is a light chain. So if you want to express this as a one protein, it's a little difficult and inconvenient to make it. How about this? If you connect those two motifs by connecting the two peptides together, so this is called as a single chain FV, then with a single peptide structure, it can mimic the variable site of antibodies. Okay, we have studied about the antibodies so far. Now, can you guess how big is the antibody? Well, molecular weight of antibody is 150 kilodalton. And how about the physical size? The height, weight, thickness? Can you guess?